So I will be giving a brief overview of my PhD project, which is mainly about the genomic variation within wild dogs from the Kruger National Park, and also about their susceptibility to Mycobacterium bovis infection, which causes bovine tuberculosis in this species. African wild dogs have experienced significant population declines and are now classified as endangered, with a global population of just over 6,000 and only 450 in South Africa. These remaining fragmented populations are expected to be inbred and have reduced genomic diversity. And this poses a significant threat to the survival of this endangered species, as it can limit their ability to adapt to challenges such as changes in the environment or to the introduction of diseases that results in their local extinction. Conservation programs address this threat by using tools such as translocation of animals between isolated populations to maintain genomic diversity. However, decisions are based on knowledge of the expected genetic relatedness of animals that is based on observed breeding and the identification of the offspring produced. And this can be difficult in the case of elusive wildlife species. Additionally, the possibility of disease transmission to new or healthy wild dog populations is not considered in these conservation programs. Based on observations in the Kruger National Park, KNP, wild dogs have a high apparent susceptibility to Mycobacterium bovis infection, which causes bovine tuberculosis or BTB, as they develop acute and chronic BTB. And advanced disease and mortality in young wild dogs have been observed, which highlights the large impact of this disease in the species. The presence of BTB in wild dogs has a significant conservation impact, since as it is a controlled disease, it can limit the use of translocations between populations for genetic management. New techniques such as whole genome sequencing can be used to gain insight into disease susceptibility or resistance, as well as provide definitive measures of relatedness to inform conservationists to make the most effective and informed decisions regarding translocations of wild dogs. So what we wanted to do is we want to know what the genomic diversity is in the wild dog population within KNP. So we did this by performing whole genome sequencing on 72 wild dogs. 22 have already been sequenced on the BGI-seq 500 platform, and 50 will be sequenced on the MGI-seq 2000 platform. These platforms are almost identical, and the reason the sequencing was split up was because we did the 22 for my MSc and added the 50 for my PhD. And then we mapped the genome. We will map the genome to a high-quality African wild dog reference genome and perform variant calling and filtering. And then we will estimate the average minor allele frequency and the heterozygosity, as well as calculate the relatedness as proportion identity by descent between individual pairs. And then we will identify runs of homozygosity for each individual wild dog to get an indication of the level of inbreeding. So currently all 72 wild dogs have been sequenced. The sequencing of the 50 wild dogs was completed very recently, and therefore the level of genomic diversity has been determined for only the 22 wild dogs. Our initial results revealed low genomic diversity based on basic indices such as minor allele frequency and heterozygosity, indicating that the rare alleles have been eliminated as a result of drastic population declines. The relatedness analysis revealed that 3% of the cohort were first-degree related, and the majority of the cohort were distantly related, and only 13% were completely unrelated. And this is concerning, as most of the wild dogs originated from different packs, so we expected that most would be unrelated. When considering conservation strategies, ideal candidates to include in translocations or reintroductions would ideally be unrelated, as it would maintain the genetic diversity. And then we also looked at inbreeding based on runs of homozygosity. In total, 54 homozygous runs were identified with average length ranging between 1 and 1.5 megabase pairs, which is considered to be relatively short, and this indicates that the inbreeding occurred between individuals in the distant past. And then currently the remaining 50 wild dog genomes have been mapped to the reference genome and variant calling is underway. So very soon we will be able to determine the genomic diversity for the entire cohort. And then what we also want to do is to use the whole genome sequence data to determine or to investigate evidence for genetic susceptibility to M. bovis infection in this cohort based on interferon gamma release assay and mycobacterial culture results. We will do a family-based association analysis to try and identify significant differences in allele frequencies between infected and uninfected individuals. This analysis allows for related and unrelated individuals to be included in a single analysis. And then we want to use this research, or this research will be used towards identifying relatedness informative variants and variants associated with susceptibility to M. bovis infection so that we can develop a high throughput genomic assay which would allow rapid and cheap screening of wild dogs from other populations to assess relatedness and disease susceptibility for conservation purposes. And this will be valuable to identify the best candidates for animal translocations that are intended to maintain genomic variation and decrease disease transmission. Thanks very much for listening.
Thank you so much, Christina. Before I go on to the other questions, I just want to know, how did you obtain the samples?
Oh, sorry, is my screen not showing anymore?
No.
Okay, so my supervisor is a vet in the Kruger National Park, and the state vets are also involved in my project, and they have been monitoring the wild dogs in the Kruger National Park for very long and have been obtaining samples for years and putting it in a biobank. So whenever they see a wild dog in the park, they would chemically immobilize them and then draw blood, get oral swabs and all kinds of things, hair, for future studies. So yeah, very lucky.
Okay, yeah, that is really great. Natasha asks, how will you correct for batch differences due to using two different sequencing platforms?
So I don't actually have a plan for that yet, but considering that the sequencing platforms are basically identical, I don't think it will be a problem, but obviously it might be a limitation of the study. But yeah, I can't answer you at the moment, but I'll get to work on that.
Okay, thanks. Peter asks, where is the sequencing happening? He knows MRC has an MGI 2000, but they are not that common in South Africa.
Okay, so the 22 were sequenced at the BGI Institute in Beijing. I think it was 2 years ago. So we had to send the samples all the way there and then get them back. And
Sure.
then this year we sequenced the 50 across the street at the SAMRC. Yeah, so it was just here.
Okay. And final question from Caitlin. Will you look at moving in wild dogs from other parks to increase diversity, or else outside of South Africa, should there not be sufficient diversity within the park?
So I don't know yet, but there are other wild dog populations within South Africa, which are quite substantial and viable and self-sustaining. So I think when that has to happen, it will first be with other wild dogs within South Africa, and then probably over a couple of years when it's not working out anymore, it would, yeah, go over to the overseas wild dog. Oh, not overseas, in the rest of the country.
Okay, thank you.